Commercial Applications of Plant Hormones by Sujal Desai, Shaz Farid, Seher Khader, and Kyle Sledge.
There are two basic types of plant hormones. The first, natural hormones, occur naturally inside the plants in small concentrations and affect the physical qualities of the plants. The other, synthetic hormones, are man-made and produced in much higher quantities, usually to increase the yield of farmers or to kill unwanted organisms. There are 5 major natural hormones. Abscisic acid promotes protein synthesis, alters dormancy and maintenance in the seeds, and inhibits stem growth. Auxin promotes stem elongation, can promote or inhibit abscission of flowers and fruits, and can also alter the fruit and its ripeness. Cytokines can promote cell division and also promote lateral growth. The final two are ethylene, which stimulates shoot and root growth, abscission of leaves and flowers, and can alter the fruit, and gibberellins, which promote stem elongation, enzyme production, and germination. There are also major synthetic hormones. 2,4-D-auxin can increase crop yield and minimize weed growth. Maleic hydrazide can inhibit cell division and produce male sterility in the plants. Cycocel is a growth retardant and also a herbicide, and ethifon, which can maximize growth, abscission, and fruit ripening.
Plant hormones are used in the food industry to help ripen fruit, increase size, and produce fruit without seeds. They are also used to spray on unpollinated flowers so that they can grow without having to use fertilization.
One specific application our group looked at was a study on the use of the synthetic hormone ethifin, which is used in commercial farming to speed up ripening. It is sprayed on plants, which then converts the hormone into ethylene. Ethifone is used in the ripening process of plants such as coffee beans, cotton, apples, grapes, peaches, and more. The experiment looked at the effects of ethifin on uba mangoes in Brazil. They took 25 mango trees and sprayed 5 trees with 250, 500, 750, and 1,000 milligrams per liter each of ethifin, with 5 trees used as controls with no ethifin on them. They harvested 5 mangoes every day till their normal harvest day and observed the color changes and firmness to see how much effect ethifin had on ripeness and firmness of the mangoes.
After the final harvest on the 6th day, scientists observed the changes that had occurred in the mangoes. They found that mangoes treated with ethifone became a darker red-orange color as the concentration of ethifone increased. They also found that the speed at which this color change occurred also correlated with an increase in ethifone concentration. Another feature they looked at was firmness. Higher concentrations yielded softer fruit, indicating that treated plants ripen quicker. The only anomaly observed was that the trees treated with 1,000 milligrams per liter of ethifone ripened too early. This caused them to fall from the tree before they could be harvested, and therefore had a negative impact on what would have been the total yield. The results of the study can further be examined in the following 2 graphs. The first graph shows a comparison of the number of days of treatment with the hue angle, and the second graph shows a comparison of the number of days of treatment with the firmness of the fruit. The hue angle is a scale that is used to determine how bright the color of a fruit is. As you can see from both graphs, any plant that was given treatment resulted in either lower firmness or a darker color than its control. Scientists ultimately concluded that treating with 500 and 750 milligrams per liter of ethifone ultimately resulted in plants that were the best colored and firmest, and therefore best for commercial success. One big concern is that hormones such as ethifone do have an impact on human health. And ethifone is a very commonly used herbicide. It's used in a lot of foods that we eat and items that we use. But studies have not really shown any adverse effects of it. Several studies have been done on mice and humans, and in these cases, both subjects were inserted or injected with small amounts of the hormone. They were then tested for carcinogenic, reproductive, and toxic effects, but none were found. The only change that did occur was in human plasma cholinesterase, which is an enzyme used in neural function. But scientists found that after 15 days of not applying the hormone again, these effects were reversed and levels returned back to normal.
Farmers are utilizing plant hormones to stimulate the growth of their crops. Plant hormones today are being used to enhance the taste of certain foods that come from crops by using artificial flavoring. They also influence plants' life cycles and increase the growth of plants.